So our final slammer before the break is Charlotte Hill. Charlotte is a final year PhD student from the Cellular and Molecular Neuroscience group in the School of Pharmacy. She's an electrophysiologist, which is the finest type of science, because I'm an electrophysiologist, and her research focuses on finding a mechanism of action to explain why some compounds from the cannabis plant are anti-epileptic. Outside of her PhD, Charlotte plays a variety of string, woodwind and brass instruments, and after completing her PhD, she intends to teach herself a few more. Charlotte's slam is entitled, How Cannabis Can Treat the Epileptic Brain, and she's asked me to introduce it thus. Some non-psychoactive compounds from the cannabis plant can be used to treat the serious brain injury or disease, epilepsy. But we still don't know how these compounds work as anti-epileptic drugs. We still don't know how these compounds work, full stop. Understanding the ways in which brain cells communicate with each other and how this is changed in the epileptic brain could be the key to understanding how some cannabinoids have anti-epileptic properties. This slam will illustrate how Charlotte is investigating this, so please listen very carefully and follow Charlotte's instructions. I'll invite Charlotte onto the stage. I'm going to move the mic across, and again, Charlotte's time will start when Hilary rings her bell.
Okay, thank you, Gary. Hello, everyone. I'd like to start off by saying that there's over 500 compounds in the cannabis plant, and approximately 100 or so are unique to the plant itself. And these are what we call cannabinoids. Now, there's only one cannabinoid from the cannabis plant which is associated with the euphoric effects that you would associate with taking cannabis. And of the other 100 or so, there's at least 2 that we know of which have anti-epileptic and anti-convulsant properties. Now, as Gary said, we still don't know how these work as anti-epileptic drugs. And as a neuroscientist and an electrophysiologist, it's my job, or it has been my job for the last 3 years, to try and work out how on earth these compounds are anti-epileptic. Now, as an electrophysiologist, I record from individual brain cells, but also a whole population of brain cells in a network. And so I've been talking about these brain cells, and basically most brain cells are called neurons, and they communicate with each other by sending signals from cell to cell to cell. Okay, so the gap between these cells is known as a synapse. So imagine this gap here, it's a very small gap, is a synapse. And now imagine that I'm a presynaptic cell, and I'm going to send a signal, for example this, across the synapse or across the void to postsynaptic cells, i.e. you guys in the audience. Okay, so now just to get you all warmed up, I would like you to clap when I send a signal. So don't clap at the same time as I send the signal. I will send the signal and then you clap. Okay, so everyone join in. Okay, awesome. Right. Okay, so in the brain you have 2 main types of neurons. You have excitatory neurons, which send excitatory signals, and also you have inhibitory neurons, and they inhibit the excitatory neurons. Okay, so they control the excitatory signals. Now, in the part of the brain that I study, I have 2 main types of neurons. So I've got pyramidal cells, which are excitatory, and they're called pyramidal because they're triangle-shaped. And the inhibitory neurons are called interneurons, and that's because they sit in between the pyramidal neurons and they control them, so they inhibit their signals. Oops, sorry. Well done. You're learning well. Right. So, do we have any couples in the audience tonight? Give me a cheer.
Yay!
Yeah. Awesome. Awesome. Going very well. Okay, right, in your couples, I would like you to decide which person will be a pyramidal neuron and which person to be an interneuron. Okay, so the interneuron could be the more dominant person, maybe. Okay, everyone else, that's you included, Tom, because we're not a couple at the moment. I'm not sitting next to you. Whoops. Right, everyone else, you are pyramidal neurons, okay? So does everyone know what they are?
Yes.
Yeah? Okay, so the pyramidal neurons, give me a cheer. Yay! Interneurons, give me a cheer. Yay! Awesome, fewer interneurons, that's good. Okay, right, so when I start sending signals, I want just the pyramidal neurons to start clapping, okay? When I send signals. Interneurons, I want you to try and stop your pyramidal neuron from clapping by any means possible. Seriously, no drinks are to be spilled, by the way. Okay, so let's get going. So, awesome. Right. Okay, so just imagine that that is the situation in a non-epileptic brain. Okay, so the excitatory signals, even though there are still a few that get through, they're mostly under control. So I found that in an epileptic brain, when I looked at the inhibitory currents or inhibitory signals by themselves, I found that there were far fewer in an epileptic brain. And this tells me that there's either fewer interneurons in an epileptic brain, or that the interneurons are still there, however, they're just not having as much influence over their pyramidal neurons. Okay, so now I want all the pyramidal neurons to clap when I start sending signals, but interneurons, you can no longer interfere with these signals, okay? So, okay, so that's just, so that's what I found, like fewer inhibitory signals. But when I looked at the excitatory signals in isolation, I found something very interesting that happened. Okay, so follow my lead. Basically chaos, you couldn't make out a single rhythm really. And this is typical of what a seizure is like. There's just excitatory signals going off everywhere, there's no control, and that is a seizure. Okay, so now when I apply my non-psychoactive cannabinoids, I also found something that sometimes happens, and it was also very interesting. So again, follow my lead. Cool, thank you. You've been a very good audience.